Hello, everyone. Welcome to BioNOW, the weekly webinar series where we discuss health, wellness, and longevity. I'm your host, Alina, and this week's webinar we will be exploring the life behind the curtains of the laboratory. Our speaker for this week webinar is Dr. Kirti Dubli, an innovative thinker with research background in bacterial communication happening at the molecular level. She completed her PhD in the biotechnology and has remarkable contributions in understanding bacterial gene expression associated with urinary tract infections. Aside of that, she has expertise in the plant phytochemistry and drug isolation. She involved in the developing, improving, and assessing the quality of the assay used in the laboratory. Very fascinating even today. So, before we proceed, I would request all the participants to adhere to the following rules. First, please keep your microphone muted at all times as a background noise can be disruptive. Second, use the chat box if you have any concerns or questions or face any technical issues. We want to inform that this webinar will be recorded and shared on various social media platforms for the learning purposes. I also want to mention that your feedback is valuable and will help us improve. So please feel free to share it with me at the end of the session, and I will appreciate it very much. Without the further ado, please join me and welcome Dr. Kirti Dubli to learn something exciting today. Over to you, Dr. Kirti.
Good evening, everyone. I'm Dr. Kirti Dubli, and the topic for today's webinar would be success story from bench to reports. Let us begin with the quote from Stephen Hawking saying, "However difficult life may seem, there is always something you can do and succeed at." Talking of molecular level, thinking of something called as DNA, decoding genetic information was all like a dream past few decades, but science has made it possible. Today, all the information that we need for an individual is present in his genes. My genetic and microbiome testing provides insights into person's health. It aids in focus on quality and health equity and aids in better determination for the treatment processes for the individual. Let us begin. Before we proceed with the details of how all the things work behind this entire process, let us understand some basic terms. Let us begin with cells. Now, cells are the basic structures of all living organisms. They provide structure for the body, allow this body to consume or utilize nutrients, and are responsible for different functions of the body. All the cells group together and perform a uniform function in a tissue. Different tissues group together to form an organ which is responsible for a specific function again. Different organs together form the organ systems, and different organ systems comprise a complete human body. Within a cell, you also have a structure called as nucleus or cell nucleus. Now, the cell nucleus is a membrane enclosed organelle which contains the chromosome, which is responsible for the DNA. Now, what are chromosomes? Let us understand that. Chromosomes are structures that look like a thread and are present inside the nucleus. These chromosomes, as you can see from the picture, where the DNA molecule has unwound from the inside the chromosomes, are nothing but the DNA. Now, chromosomes are not just DNA alone, but they also contain protein molecules which are called histones. It is upon these histone molecules that the molecule of DNA coils and coils all together. There is coiling and supercoiling and further levels of coiling in order to fit this long molecule of DNA into a very tiny structure called a cell nucleus. In short, we can also say chromosomes are actually responsible for what a person is. As far as human beings are concerned, humans have 23 pairs of chromosomes or 46 chromosomes as such. Now, these chromosomes are, as I earlier said, are made mostly of DNA and histones. It is this DNA which is the genetic material or is also responsible for the genetic code of an individual. This DNA is the complete instruction manual for an entire human being. Every DNA is unique for the human being. So let us understand what is genes and how are they actually present, correlated with DNA and the genome. So genes are the fundamental building blocks of the body. They are group of sequences on the DNA. Now these genes are responsible for making proteins. These proteins, which are produced from the genes, are responsible for different functions in your body. They can act as enzymes, they can act as hormones, they can act to provide structural strength to your body. They are responsible for pigmentations, they are responsible for your hair, eye color, and many other features of human body. Certain genes are also responsible for coding specific RNA molecules which have some specific functions. Now, let us understand the next important term, that is genome. Now, what is genome? Before we go on to genome, in this following picture, you can see that you have chromosomes. Within chromosomes, you have DNA, and this DNA has units called genes on it, which is responsible for proteins. Now, let us understand what is genome. So when we talk of genome for any individual, it is a complete set of DNA instructions in the cell, and as in case of humans, it refers to 23 pair of chromosomes located inside the nucleus of the cell. This genome is required for the functioning and development of the individual right from the stage of single cell, which is zygote. Coming to exomes. Now, exomes refers to the coding regions of the DNA. So when we talk of chromosomes and DNA and genes all together, not entire genome is responsible to code for proteins. Not entire genome has genes. It is only 2% of the entire genome which is responsible for coding proteins, and these coding regions of the genes are referred to as exomes. So let us begin to understand the process which is involved in sequencing and decoding of this beautiful molecule, mysterious molecule called DNA. Well, this was a mystery in the past, but not mystery any further with the advancing scientific technology. So today, most of the sequencing which is done in the labs is based on next generation sequencing. Now, this next generation sequencing, also referred to as NGS, is used for studying genetic variations which are associated with different diseases like cancers, diabetes, different kinds of autoimmune disorders. Besides, also being used for studying or analyzing different biological phenomena. This technique came into being commercially in 2005 and was also referred to as massively parallel sequencing. Well, this was called so because it enabled the sequencing of many DNA strands at the same time instead of one strand at a time or singly, which was the norm of traditional Sanger sequencing. So today, what technique which is mostly followed is referred to as shotgun sequencing. This sequencing involves breaking up of large genome into smaller fragments of DNA, and these fragments are then individually sequenced. Once sequenced, a computer program looks for the overlaps within these sequences, and these overlaps are reassembled to form long fragments and in correct order to reconstitute or form the entire genome. So, let us go into details or proceed into details of sequencing associated with whole genome, exome, and microbiome. Let us understand each of these terms specifically. So, let us begin with whole genome sequencing. So, the whole genome sequencing refers to sequencing overlapping fragments of DNA in parallel. And as I told you, the computer program is responsible for assembling these smaller sequences or fragments into larger contexts and eventually an entire chromosome. Now, This whole genome sequencing is a very detailed and a comprehensive method for analyzing the complete genome. It is very much instrumental in identifying different kinds of diseases which are inherited. It can also be used to study the progression of diseases like cancer, and can also aid in tracking disease outbreaks. This technique has its own advantages as it is responsible for providing high resolution. As this technique of whole genome sequencing involves detailing each and every base, it gives you a complete view of the genome. It is also responsible for capturing small as well as large variants, which can be missed when we have a selective approaches which are used. It can be used for identifying certain causative variants and follow up in various gene expression studies, and utilize it for understanding different regulatory mechanisms at molecular levels. Besides this, it also delivers or produces large amounts of data in short amount of time, which allows an assembly of complete genome and understanding the entire genome from perspective of a single disease or condition as such. Now, coming to whole exome sequencing, it is another form of next generation sequencing which is also commonly used. Now, this whole exome sequencing is primarily refers to sequencing those regions of genome which are involved in protein coding. As I previously explained in the slides, exome comprises of nearly 2% of the genome, while rest of the 85% of the genome is not involved in protein coding regions. So when human exome is studied, it allows in easy identification of disease related variants, because most of the variants are concentrated in the exonic regions. Moreover, as less of the genome has to be sequenced, it is a cost effective alternative when compared to whole genome sequencing. Besides, enrichment techniques can also be utilized to identify different coding variants for specific applications, and this can be used in different fields like population genetic diseases, and cancer studies. This technique also has its own advantages as it leads to identification of variants, specific variants in the exonic regions. It provides comprehensive coverage of the coding regions of the genome. As previously explained, we are just going to, as exome sequencing covers only small percentage of genome, it requires only 4 to 5 GB of sequencing per genome when compared to nearly 90 or sometimes more than 90 GBs of data for the whole genome. Moreover, it produces a smaller, easily manageable set of data which aids in faster analysis of the of the exomes, and better providing targeted approaches for development of treatment pathways. So the next kind of sequencing which is commonly associated is with respect to the microbiome. So before we understand what is the sequencing of microbiome, we need to understand what microbiome is. So, the human microbiome comprises bacteria, viruses, archaea, and eukaryotes, which can involve different kinds of parasites, fungi. All these organisms reside within or on the outside surfaces of our bodies. These organisms are capable of impacting human physiology, both in case of healthy conditions as well as disease condition. Moreover, they also lead to contribution in enhancement or impairment of different immune functions as well as metabolic functions. Now, microorganisms are omnipresent, as we all know, and so they colonize various sites on the human body as well as inside the human body. And these microorganisms are capable to adopting specific features of each niche. For example, if we are referring to microorganisms in the gut, most of them are facultative anaerobes. If we are referring to microorganisms in the respiratory tract, nasal tract, skin surfaces, most of them are strict aerobes, depending on the habitat where they are found. So the organisms which are indigenous to human body are very well adapted to human immune system, and as they are interacting with the human immune system, they are not considered as pathogens, or they are not always very harmful to human beings. But an alteration to these the balance of these microorganisms can be detrimental to human health and can lead to dysbiosis. This dysbiosis can result in disease pathogenesis as well. So this dysbiosis increases the susceptibility of humans towards any specific infection, and moreover it can also be dependent on what kind of anatomical site it is present at. The human microbiome is extremely diverse and is responsible for specific metabolic functions and activities. So, it is also equally important to sequence this kind of microbiome to understand, because it has a major contribution to human health and well being. So now let us proceed ahead and understand what are different microbiomes which are present in the human beings and how they are important to us. So the first microbiome is the gut microbiome. This microbiome plays an important role in the development and refinement of immune system. It also provides protection against pathogens, aids in digestion, and is involved in nutrient metabolism. The next microbiome that is commonly found, or that is commonly studied in across the literature, is oral microbiome. It is the microbiome of the oral cavity. This microbiome is refers or rests basically within the biofilms which is found inside the oral cavity, and is responsible for maintaining the health of the oral cavity. Oral microbiome is equally important as the diversity of oral microbiome can impact gut and overall health. The next type of microbiome is the skin microbiome. So, skin microbiome is the microbiome which is present on the skin surface, and this microbiome is highly resilient because skin is the organ of the body which is exposed to the external environmental conditions, which can be extreme heat, high humidity, low humidity, it can be lower temperatures, and different kinds of chemicals. So, this microbiome has a vital role in defending the disease causing microorganisms, as it is also responsible for primary immunity. It boosts the barrier protection against the pathogens and also aids in immune defenses. Last but not the least, the most widely studied microorganism is the vaginal microorganism. It refers to the microbiome present in the female genital tract. It is a mix of this microbiome is a mix of communities of bacteria, of which the most dominant species present are Lactobacillus crispus. These microorganisms are involved in providing protective function for the female reproductive health. So now let us proceed ahead and understand what are different challenges mitigated in some of the processes which are commonly utilized in sequencing processes. Here, I'll just be focusing on two instead of talking on everything. So first, we'll discuss what are the problems associated with whole exome sequencing, and what are the common solutions which are followed across the globe to deal with these problems. The first and foremost common problem is hybridization. Now, this hybridization refers to probe binding to specific regions so that the sequences are so that the sequencing process is efficient. Now, many times the hybridization of probe is not efficient, and this can be solved by using correct ligation conditions, like using optimum temperatures, optimum period of incubation, no high heat, no low heat, optimized conditions for binding of the probe. The next common problem is post hybridization yield. This post hybridization yield can be problem of this post hybridization yield can be solved using careful processes, optimizing each and every step, and breaking down the steps to simpler versions so that the better results or improved results can be obtained. The third most common problem which is associated with WS is the product yield. This product yield can be optimized by using high volumes of initial reactants, besides using individual products for library processing, which will aid in better whole exome sequencing. The next technique that I would be discussing, where the problems associated are skin microbiome. So, talking about skin microbiomes, the first and foremost problem that is commonly encountered across the globe is sample collection and storage. The best possible solution for this problem is to analyze different collection sites and optimize the duration of storage process so that the yield of the DNA is not lost. The DNA is successfully recovered from the microbiome which is found on the skin. The next problem which is associated is sample processing and extraction because of low bioburden. As I previously mentioned, skin is an organ which is exposed to external environment, and the cells which are found on the skin surfaces are highly resilient. Hence, it is very difficult to break these resilient cells. The only solution is to break these cell walls of these resilient cells and utilize and remove various enzyme inhibitors which prevent DNA isolation and further downstream processing. The last problem which is encounter is with library preparation and sequencing. The best possible solutions that can be utilized is optimizing DNA input, removing host DNA so that the microbiome DNA is specifically studied, and accurate capture of microbiome and utilization of it through the computer program to analyze the data. So let us proceed ahead and understand what happens after this process, after sequencing. The sequencing is generally followed by report generation. So report generation across the globe comprises of following features. Let us begin with the summary. The report generation gives you a brief description of the microbiome composition. What is the microbiome composition of the specific organ and how it is correlated with different diseases and condition. Besides, it also provides a knowledge of presence of beneficial bacteria within the microbiome as well as the pathogenic bacteria which are present within the microbiome. The microbiome reports also compare the microbiome sample of the sample with that of the healthy subjects in the form of heat map, wherein a accurate percentage abundance can be compared with respect to the healthy data set and the test sample. Microbiome reports also talk about different species or top species which are found in microbiome and deal with different significances which are associated with these microbiome in impacting the human health. Microbiome reports also talk about different pathogens, commensals, and opportunist bacteria which are found in the microbiome by comparing it with the microbiome database from the healthy data set. Besides, these databases also give you an idea of the significance of how these organism pathogens, commensal, or opportunistic bacteria can affect the human health. Besides this, the microbiome reports also focus on different major outcomes. The first one is comparison with healthy reference cohorts. Microbiome reports compare the sample data set with the healthy reference cohort to give a fair to compare to generate a report in comparison with healthy subjects which are classified or which are selected on the basis of stringent inclusive and exclusive criteria. Microbiomes are generally comprising of four major categories of bacteria, which are referred to as firmicutes, bacteroidetes, actinobacteria, and proteobacteria. Microbiome reports focus on the presence of these major groups of bacteria in the test sample and determine their significance with respect to the test sample. Microbiome reports also talk about something called as Shannon diversity and OTU. Now, Shannon diversity is for evenness, It helps us to determine the diversity of species within an ecosystem or within a sample. Similarly, OTU is for richness. It refers to classification of bacteria on the basis of sequence similarity of the marker genes. The next feature which most microbiome reports focus on is antimicrobial resistance. In the present scenario, we know that the treatment of infections basically focuses on usage of antibiotics. But in past few decades, it has been seen that due to rampant use of antibiotics, there is a phenomenon called antibiotic resistance, which is arising in the bacteria. And sometimes this antibiotic resistance is seen for multiple drugs, leading to rise of multiple drug resistant bacteria. Now, this antibiotic consumption can be detrimental to the human health, as well as it can lead, as well as studies have shown that it can lead to decrease in microbial biodiversity, as well as alteration of the microbiome of the organ concerned. Besides giving rise to multiple drug resistant strains like MRSA, as we see, as well as the multiple drug resistant strains seen in diseases like tuberculosis. One such example is also Clostridioides difficile, which is responsible for diarrhea. Studies have shown that it has reduced susceptibility to drugs like metronidazole. So, as microbiome reports focus on antimicrobial resistance genes, it will these kind of studies will allow healthcare providers and the policymaker to design policies for the better for providing better healthcare to the individuals. The other feature, which is commonly associated with most microbiome reports, is the detection of virulence factors or proteins. Now, what are these virulence factors or proteins? So, virulent factors or proteins are those components of bacteria which help in establishing the bacteria within the host organism and is responsible for pathogenesis. So, some of the commonly seen virulence factors or proteins are endotoxin, enterotoxin, adhesive flagellae, O antigen, which are found on the surface of the bacteria. You have siderophores, which are produced by bacteria, which are also commonly seen virulence factors. You have capsular antigen, which is seen on the bacterial cell surface. You also have certain surface appendages, which help in better adhering of the pathogenic bacteria to the host cells. Now, all these virulence factors aid the bacteria to attach to the host cell surface, infiltrate the cell surface, like it traverses through the epithelial layer of the tissue, enters the cell. Besides that, it also attacks intracellular components of the cell, and many a time it helps in dodging the immune or defense system of the host, in turn leading to establishing of the disease inside the host organism. So microbiome reports basically focus on detecting these virulence factors so that it can give an idea of how these virulent vectors can be dealt with, so that the healthcare professional can provide a specific targeted treatment to the individuals or the subjects. So as we can see that these microbiome sequencing studies are extremely important, and most widely studied microbiome is the gut microbiome. Gut microbiome has a huge impact in human lives. Gut microbiome is the microbiome of the gut. It performs plethora of function. So let us see what are the different functions it can perform. According to literature, studies have shown that these gut bacteria of gut microbiome are capable of breaking down complex carbohydrates, dietary fibers. They also produce molecules called as short chain fatty acid or SCFA. This is an important nutrient. This nutrient is produced as a byproduct. They provide enzymes which are necessary to synthesize vitamins like B1, B12, B9, and K. Besides this, these microorganisms are also responsible for training immune system to differentiate between unhelpful and pathogenic forms of bacteria. Gut gut comprises or gut, and when we talk of gut, it is one of the largest immune system organ, because most of the nearly 80% of body's immune cell are concentrated in your gut. These cells are responsible for clearing the pathogens which are passing through the gut every day. Besides that, literature has also shown that gut microbiomes can impact the nervous system, because they are responsible they actually produce certain molecules which are responsible or for stimulating neurotransmitters, like production of neurotransmitters like serotonins, and these serotonin molecules are known to play an important role in the well-being of the nervous system. Moreover, studies have also shown that gut microbiomes can interact with endocrine cells of the gut lining. These cells, also referred to as enteroendocrine cells, are nothing but one of the cells which can synthesize different hormones and regulate different aspects of metabolism. So, as we have seen, gut microbiome is very, very important. We can see the gut microbiome is extremely huge, and when we see the heat map of gut microbiome for large number of subjects, we can see that it produces a very interesting heat map with lots of species which can be seen on the y-axis. Studies have also shown that the gut microbiome is capable of impacting the body mass index. Certain bacteria like Faecalibacterium prausnitzii, Phaeola vulgatus, Blatia obium, Anerostypes hydrus are play an important role um, in a BMI of an individual, and also affect the conditions like obesity. Studies have also shown that gut microbiome is linked with the cancer. In the following Venn diagram, one can see that how the gut microbiota of healthy subjects is comparable to those of patients with breast cancer, skin cancer, colorectal cancer, and liver cancer. The 26 of the study conducted, 26 microorganisms are exclusively found in healthy subject, while 53 are common in all the four categories, which are healthy breast cancer subject, skin cancer subject, colorectal cancer subjects, and liver cancer subject. So, gut microbiome is also dependent on the type of diet a person consumes, and studies have shown that the gut microbiome is related with the amount of healthy microbiota and pathogenic microbiota which is found. Studies have indicated that non-vegetarian food diet has contributes to seven common pathogenic microbiota with when compared to the vegetarian microbiota. But when we speak of individual contribution of non-vegetarian diet, it contributes six pathogenic bacteria when compared to vegetarian diet, which contributes one one bacteria to the entire group. So, at the end, I would say that at the end, I would just like to say that when we talk of microbiome sequencing, this field is very endless. And as science is progressing further, there is nothing called as endpoint. We should always remember that success is not final and failure is not fatal. It is just the courage to continue that counts, as said by Winston Churchill. So we must all remember that the progress that we see today is not just an effort of a single person, but it is effort and success story of many researchers who have spent day and night continuously to work on this process and develop these processes so that they can be used by different researchers and healthcare providers to provide best possible treatment or health care to the individuals, so that they can live a healthy life and have a healthy mind. Thank you.
Thank you very much, Dr. Cutie. Amazing talk. We are grateful for you to sharing your wealth of knowledge with all of us. I'm definitely more informed and aware about the science behind the next generation sequencing and the microbiome analysis. Dr. Cutie will now take questions from the audience on discussion we've had today. Please use your chat box for asking questions or request to unmute if you would like to use your microphone. Okay, we have one question. So, Dr. Kirti, can you better illustrate your point on use microbiome investigations for the patient management? Or, for example, vaginal microbiome?
Yeah. Studies conducted by, yes, as Dr. Raja Singh said, yes, it is very, very, very much important for investigations, because different studies have shown that vaginal microbiome also impact the well-being of the individual, especially in case of preterm birth. Studies conducted by different scientists, specifically Fetwise et al. In 2019 has shown that decreased abundance of bacteria like L. crispatus and increased abundance of certain species like BVAB1, Snedia or Aminis, and TM7H1 belonging to Prevetola has resulted in more incidences of preterm birth. So yes, definitely these kind of studies are aiding healthcare providers to work in a better way to provide healthcare to the individuals.
So, thank you very much for the answering the question. We have another question. How quickly do you feel the microbiome can change?
Yes, studies have shown that microbiome can change as I can specifically in gut microbiome. The gut microbiome studies have been indicated that they can change as quickly as within a week to 2 week after changing the diet.
That's a very rapid change, and it can contribute to the very good improvements if we will make sure to pay attention to our microbiome. We have one more question. Can microbiome be used for prediction or prevention of disease in fetus in a newborn? I believe that's
Yes, yes,
something that can.
yeah. Studies are actually going on in this particular aspect of vaginal microbiome, and yes, there are studies which are indicating like the presence of certain species of bacteria is responsible for some diseases which can be seen in newborn or fetuses. And yes, when these kind of studies come over, healthcare providers are actually able to work on it and provide better kind of solution to the individuals. There is not much study into this, but yes, studies are going on into this, but commercial applications are not that wide.
Thank you. There is one more suggestion and a question. So, thank you for the talk. Besides the shotgun sequencing, which other technologies are available for the microbiome sequencing, and what are the advantages of shotgun sequencing?
Yes, there are many other technologies available for sequencing. The most commonly, the labs that we use are 16S RNA sequencing, but these kind of sequencing give you idea about the bacterial genuses, but not about specific species. And talking about shotgun sequencing, it is definitely advantageous, as I previously said. Here, in case of shotgun sequencing, the large DNA fragments are broken down into smaller fragments, and it is these smaller fragments which are then sequenced and then laid together based on overlaps to form a larger context to give rise to entire chromosome. So as smaller fragments are getting sequenced, the process becomes shorter, because when we are doing the sequencing using a traditional way like Sanger sequencing, the entire stretch of chromosome has to be sequenced and analyzed. But when we have shotgun, we have shorter fragments of DNA which are sequenced, realigned, and we have the data in a shorter amount of time when we compare it with Sanger sequencing.
So, one more question. Which type of the sequencing would you recommend for the clinical purposes?
Well, definitely something like shotgun sequencing, because if it does the sequencing process in a shorter amount of time, definitely the shorter amount of time is not just like few hours, it takes days, but this amount of time is definitely less than what we do it in the traditional Sanger sequencing.
We'll probably give a couple seconds to the audience for the last questions. So, there is additional comment for the previous question. Would shotgun followed by the 16S help or be persuaded?
Well, I believe that instead of going for 16S, one can actually directly go for shotgun sequencing, because it will straight away give you a specific detail rather than 16S, which will give you a generic detail talking about just genus level of information. Because when we talk of genus, there can be n number of bacteria within a genus. So when we are talking of shotgun, it gives you specific details rather than the 16S sequencing.
Thank you, that's amazing. Dr. Raja, you should be able to unmute yourself now. Mm,
Okay, can you guys hear me now?
yes, yes, we can.
Thank you so much, Alina. Uh, Kirti, excellent presentation. Thank you so much. Where I was going with that question was that you said that the shotgun sequencing is better because with one sequencing approach, we'll be able to get a sense of all the organisms which might be present in that particular sample. Now, once we have that, should that be followed by a 16S if we are trying to have a better sense of uh, the genealogy or the genetics of the organisms involved, especially if we are interested in a specific organism?
Like we do have something like 16 as shotgun sequencing approach, which actually which are responsible for reading all genomic data in a specimen rather than just one specific area. So maybe we can do that. But when we talk of shotgun sequencing with 16S, they can actually help in identification and profiling different organisms. So I
Right.
think that is the way we can go ahead with.
So only in specific cases where once we have identified if we are looking for some antibiotic resistance segments of the bacteria, right? Then maybe we can go into 16S.
Uh, like both the approaches can be used, but again, I feel that as we are talking of specific thing, we should go with shotgun, because more specific analysis is always better for the better treatment provision.
Right, right. Okay, thank you so much.
Thank you for the question. Right now, we definitely have a comprehensive difference between 16S and a shotgun sequencing. Okay, that brings us to the end of this webinar. Thank you for your participation and all your questions. I would also like to take time out and thank to all of you, our virtual audience, who joined us from within Canada